Hi Hires, problem solving question from the 2019 CFA paper from the SQA, question 12. This is on experimental design. Right, our context for this one. We have an investigation carried out into the effect of increasing time of exposure of UV light on survival of wild type and mutant cells. You're given a code that's going to be used throughout, I'm assuming. Each type of yeast cell was grown in separate liquid media, 30 degrees for 24 hours, diluted and then plated out. They were then exposed to a UV light source for between 0 and 30 seconds, and the plates were incubated for 4 days at 20 degrees C. The number of yeast cells were counted. Okay, each colony grew from a single cell, they're stating. Okay, so here's our data. We've got time of exposure to UV light in seconds, so that's increasing, going down here. We have our wild type colonies and our mutant colonies, and then we have the number of each that survived at each of the doses. Okay. State an independent variable in this experiment. Note that there, for SQA being nice about this, an. So they're telling you there is more than one, not state the, but one of. And there are two, because what we have is the time of exposure to UV, and then we've also got two different yeast cells. So either of those is going to be an independent variable for this. Suggest why exposure to UV light was carried out in a darkened room. Well, this I hope is quite obvious. Basically, light contains, in terms of normal sunlight, contains UV. So if you want to actually expose it to a known quantity of UV, you're going to have a darkened room to put that in. And that's it. On the grid, draw a line graph using the results in the table, here we go, for both wild type and M. Now this is worth 3 marks. Most of the graphs you've seen so far have only been worth 2, although always a line. So what we're going to have to do, first of all, is take, this is going to be our what we changed, so it goes along the bottom, time of exposure to UV light, and the whole thing, don't miss anything out, just lift the entire title from the table, and this one is going to be number of yeast colonies. Okay, so our time of exposure is quite straightforward. We're going 0 up to 30, so 5, 10, 15, 20, 25, 30 fits very, very neatly. And then we're going up the side. Our biggest number is 400, so that looks like 100, 200, 300, 400. You do not leave these check marks empty. If they're there, fill them in. Okay, if they've been put, and if you're drawing one without that doesn't have check marks and you've put check marks in, you must fill those ones in as well. As soon as you have one of these little things to the side, you need to put a point on it, like a number. Okay, so that's going to be 1 mark. Second mark is going to be for your plot. So if we take just the wild type, in fact I'm just flagging up one that I can see straight away is going to be an issue. Because this one's going to be within a half box. So at 15 you're going to go 10, 20, 30, 40, and the 45 is going to be between the 2 points on the 2 lines on the grid. That's what they expect you to do. If you can't manage, you have to be able to get on that point. If you put it on the crosshairs above it's 50 and below it's 40, it has to be in the middle point to be the 45. Okay, so most of those are going to be on the cross. That one's not, so watch out for that. And then that's your second mark. I'm not going to draw it on here because I just make a mess when I'm trying to do it on the graphics pad, because it's not a fancy graphics pad. And the reason it's worth 3 marks is because you have 2 lines. So you're going to have to label one of the lines as wild type and one of the lines as your mutant. So either that is with a key, so you might have this at the side, or you might have used different markers for it. Either a key at the side or at the end of the line, wherever it comes to. So, well, your M1's going to be down here, isn't it? It's going to be 20 and 30. So when you've joined them up at the end of that line or on that line, you're going to put M, so it's clear. Okay, so third mark is for the labelling on the lines. Draw one conclusion for the result of this investigation. Oh, here's our results. So conclusion. Well, both of them, you can see that as this increases, so as the exposure to UV light increases, the number of yeast colonies in both of these is dropping. Okay, what you can also see is that the wild type survives higher exposure than the mutant. Okay, you can also say that at no UV that the mutants actually have a higher level than the wild type. But because this was looking at the time of exposure, you really do want to bring this into the conclusion. There's a list on the mark scheme of what's acceptable for this one. Okay, last question on this. It's quite a short question set for this one. Sunscreen lotions can protect cells from UV damage. Suggest how the investigation could be modified to test the effectiveness. Right, so using the M cells, that's mutant cells. Well, first of all, you're going to need to apply the sun lotion. So that's probably basically just a smear of it on the plate or on the surface of the plate or on the lid of the plate to prevent the UV getting to it. Okay, and then the second thing is you need a comparison, because you can't tell if it's had an impact unless you have your kind of blank. So what you're going to need is some form of without the sunscreen as well. So you want one with and one without, and that would allow you to do it. And there's your 2 marks. Right, so your SQA mark scheme, pretty straightforward. There's nothing that I think that you'll be looking at and saying, okay, I don't understand why that would be the mark or not the mark. Just a couple of things that they are being absolutely stringent on. Lines must go through all points on your graph, so they are not allowing you to just put a curve of best fit or anything on that one. They are looking for joined up dots. And everything else I think is quite clear as to what they accept. Okay, that's it.